Good afternoon, everybody. So my name is Han, and I'm an associate professor in the Human Genetics Center at University of Texas Health Science Center at Houston. So today I'm going to present some of our recent work, a reference panel-free summary statistics framework for diverse populations. So before I get started, I would like to briefly introduce some of the background information. So in a broad sense, genomic summary statistics includes any information that's aggregated from individual level data. For example, I mean, in a broad sense, allele frequency is a genomic summary statistic measure. And within the scope of today's talk, I'm going to restrict the genomic summary statistics to the narrow sense definition, which is a single variant statistics. From genome-wide association studies, usually, you can run a linear logistic or Cox regression and get their regression coefficients and standard errors. You get one of these values for each SNP or genetic marker, and then you get millions of them out of the whole genome. Or sometimes people also use the Z-scores, which is the regression coefficient divided by their standard error. So in some of the applications, in addition to the regular GWAS summary statistics, we also need the second moment information, namely the cross-variant correlation or linkage disequilibrium matrix. So this information would be very useful for us to estimate the covariance or correlation between the GWAS summary statistics, and they can be applied in a wide range of applications as long as you need to look at two or more genetic variations together. For example, we can get this LD matrix from the study samples, which is basically straightforward. Not so straightforward if you have very large samples, but a common practice is to use the ALD reference panel from the external data sources, and that approach has been very successful. So taking them together, we can do a lot of things with genomic summary statistics. We can do meta-analysis. That's a very simple application. We can do heritability, co-heritability, or genetic correlation estimation. For rare variant analysis, we can do variant set, conditional association tests. If you know the correlation between the variants that you're looking at together. And you can also do LD score regression. Basically, in LD score regression, we will need the GWAS summary statistics, and we also need the LD score. And the LD score was computed based on the linkage disequilibrium information using all the adjacent genetic markers. And in some of the multi-phenotype analysis approaches, we also need that information. And we can also do transcriptome-wide, prediscan, association analysis, polygenic risk score, median randomization. So we can do a lot of things using genomic summary statistics. However, for the traditional genomic summary statistics, there are some limitations. Namely, I think for rare variant analysis, I spent many years trying to develop computationally efficient genetic association tests for rare genetic variants. But one of the fundamental challenges is that usually a single variant test has very little power because of the low mean allele frequency. So we have to aggregate those genetic variants together in some way to test them together. And in that case, the single variant summary statistics must be combined with the ALD matrix in meta-analysis. So suppose that you have several different studies, and each of them you have whole genome sequencing data. You want to combine them together using the summary statistics. Then there are a lot of software programs that you can use. Metastar is one of the most recent, which was published in Nature Genetics last year. And all of these would require the variant sets to be predefined. For example, you can do a gene-based analysis by testing each gene. You get one p-value for each gene. Or you can do a sliding window analysis if you have whole genome sequence data. Calculating the p-value for each 2 kb, 4 kb sliding window across the whole genome. So you can do that. So this approach, if you want to do meta-analysis, you need both the single variant summary statistics, and you also need the second moment information, which is basically the covariance or LD matrix. And this generally needs to be pre-computed, before you combine them together, and they can be computationally very expensive if you're testing a very large region, for example, like topologically associating domains. You want to look at one TAD and then try to aggregate all the rare variants in that region. So you have a very big region, so you have a very big covariance matrix. And in this field, LD matrices from external reference panels are not accurate because you can imagine many of the rare variants, they are singletons or even private. So the allele frequency will largely vary, if you look at the external reference panels, even if they have the same ancestry background. So this is not the common practice for rare variant tests. So most people would actually compute them internally from the study samples. So that makes international collaborations extremely difficult. Part of the reason is that when you are coordinating a meta-analysis, you have the analysis plan written, you ask 100 studies to participate, and then later, something happened and you want to change the variant set definition, then you need to go back to each and every individual study, ask them to rerun the analysis using the new definition, and then aggregate the information together. So, if you want to do some post-analysis lookups or condition analysis, you need each and every study to do the same. So that's extremely difficult. And another challenge, came from underrepresented or unmixed populations. This happens even for common variants because it's usually difficult to find external reference panels. And a compromise is to use European reference panels. But for many of the summary statistics-based applications, we see reduced accuracy or performance when using European reference panels. So one compromise is that, for example, if you have a cohort of Hispanic Americans, so they have a three-way admixed ancestry background with a European, African, and Amerindian ancestry populations. So an alternative approach is that, so what if I add all those ancestry reference panels together as a reference panel? Would that work? Does that work well? No? We will see later. So I have some results showing you the empirical results. But the short answer is that, sometimes it works better than using only European reference panels because you have more haplotypes. But generally, it doesn't work well if you want to look at the LD because these are the mixed populations. Even though they have the same global ancestry proportions, let's say if you have two individuals who have, on the global ancestry level, 50% Europeans and 50% Amerindian ancestry, if you look at a particular genetic locus, then the ALD structure can be very different because some part of the segments could be inherited completely from a European ancestor for one individual and a Amerindian ancestor from another individual. Therefore, if you look at their local ancestry LD structure, it's very different, even though you have all the ancestry reference populations covered in the reference panel. So, and on the other hand, we also often need complex statistical models, namely linear or logistic mixed models, to handle the admixture cryptically related relatedness in this kind of populations. So that's even more complicated than a simple linear or logistic regression model, and that's computationally expensive. So the solution that we came up with was to go stochastic. What does that mean? It means that we're going to use some randomized matrix algorithm to enable efficient and flexible estimation of cross-variant correlation in the study samples. So we don't really need the reference panel. And what it does is that it can make existing summary statistics-based approaches, which were originally developed and performed extremely well for European populations, we can make them equally applicable and beneficial to underrepresented and mixed and isolated populations because now we can get the LD information from those study samples using very limited computational resources. Okay. So today I'm going to talk about two specific applications. So number one is that we can use this framework to test distant rare variant sets. I'm going to give one example, and I'm also going to talk about how to compute the LD scores. and then LD scores can be used in downstream analysis to estimate heritability and genetic correlation. So this is a general framework for whole genome sequence data analysis, before using the traditional approach. I apologize if this is not looking very well. But basically, we start with the phenotypes, the covariates, and the kinship matrix if you have related individuals. And this can also be replaced by a genetic relationship matrix if you're concerned about population structure. So we use this information to feed null model. And then we use the whole genome sequence genotypes and the null model to compute the single variant scores. And along with a variant set definition, remember that this needs to be predefined before you run the analysis. To compute the covariance matrices, this is going to be a block diagonal matrix across the whole genome. And with those information, we can do single study analysis, and we can also combine similar information from other studies doing a meta-analysis. If we do LD score regression, we need the single variant scores. We need the LD scores. LD scores were usually estimated from the external reference panel. So that's another way to use that information. So you can see that in this framework, the covariance matrices depends on the variance set definition. So if you don't have the variants in the same set, then you don't have their covariance information. So that's not very convenient, especially if you want to change your analysis plan, you're coordinating a 100 study meta-analysis. So our solution in the stochastic summary statistics framework is that, okay, first of all, we still start from a null model, and then we can generate some random vectors from the null model. And then using those random vectors, we can multiply them by the whole genome sequence genotype to get a stochastic summary statistics. And these two random vectors were generated. So for LD scores, we started from the whole genome sequence genotype from the same study sample, and then we can generate some random vectors and then to compute the LD scores. So you can see that now we can move the variant set definition. If you remember previously, it was here, right? Upstream of covariance matrix. Now we don't need covariance matrices. We can move the variant set definition here at the analysis stage. So you can decide how you want to group your variants at the very end stage, right? You can even do that on your laptop. Once you have these, summary statistics, you can play a lot of crazy things. So some mathematics. So our general model came from a generalized linear mix effect model. So this is general, and it covers a lot of situations. For example, this is a link function. If you have an identity link function, this reduced to a linear mix effect model. And if you remove the random effects, then this is going to be a generalized linear model, including logistic and linear regression. So our general hypothesis is we want to test the genetic effect beta. And for single variant tests, beta is a scalar. And for rare variant tests, usually we're testing beta as a vector. We're testing several variants together because the single variant test is not very powerful. So we will first fit a null model. You can see that this model does not depend on any genotypes. So this only needs to be fitted once in a genome-wide analysis. Our original solution, this is a GWAS paper. Which made the generalized linear mixing model-based association test computationally feasible, was that we were using a score test framework. So here R is a residual from this null model, and P is a projection matrix from this null model, and then we can basically calculate the cross product as well as the quadratic form, so that we can get the single variant score and its variance. So if you want to do a rare variant test here, the classical sequence kernel association test, which was one of the earliest rare variant association tests, the statistics, it turns out to be a weighted quadratic form of these residuals and the genotype matrices. And now we need to compute the eigenvalues from this weighted matrix. And remember that now G transpose PG is a matrix, it's not a scalar anymore. So the challenge here is that, because we have to define, in the SCAT framework, we have to define how we want to group G first. So this needs to be computed using individual level data. And this, you can see that if P is a n by n dense matrix, this has a n square computational complexity for dense GRMs. And another challenge is that we need to predefine the variant set for grouping G. And therefore, if you decide to change your variant set definition, then you need to start over from individual level data again, which is a waste of computational resources. So the heuristics that we're using is, statistically it's very simple. So this is the quantity we want to compute, right? You have a genotype matrix, the projection matrix, and this, we want to compute this V matrix. Instead of computing this quantity directly, we want to use B length and random vectors. We simulate RB from this multivariate normal distribution with a covariance matrix equal to this projection matrix, and then we assemble them into an n by B matrix R. So this is what I have. Once I have this R matrix, all I need is to do a cross product of these two matrices. We get an stochastic summary statistics matrix U instead of computing this V matrix. So you can see that there are several advantages for doing that. First of all, if the number of markers M, the dimension of this G matrix, is small relative to the number of random vectors B, then by law of large numbers, you can show that this cross product will converge to this V matrix. Therefore, when you want to test, do the variance component test for some rare variants, when you need the eigenvalues of this V, you don't really need to compute this matrix and then do the eigen decomposition. You can basically do a singular value decomposition of this U matrix to get the singular values and then take the square, which, and then you're going to get the eigenvalues. So now you can see another advantage is that without specifically defining how to group this G, we can compute this U matrix first on the whole chromosome scale, where M is much greater than B, with complexity which scales linearly with the number of variants and the sample size. So on the whole chromosome scale, even though this is no longer true because B is much smaller than M, if we want to look at some pairwise LD, it still holds, right? Because you can look at those are just the ith row and the jth row of this U matrix, right? If you want to look at two variants at a time, that quantity still converges to the corresponding element in this V matrix. So we can make use of that. This is the genetic association test using the predefined variant sets. So you can see that the LD matrices from the study samples needs to be predefined. Then you calculate this kind of block diagonal LD matrices. An updated version is the MetaStar paper, which was published last year. They use a banded sliding windows. So they basically computed all the variants within 500 kb proximity. So, this works better than that. But it still doesn't work if you have variants that are physically located more than 500 kb away. So, when people use the reference panel, there's a big trouble, right? Because, some of the variants in your study sample may not exist in the external reference panel, then you have to exclude them. So, for example, when you look at this yellow matrix, that's going to be smaller than what I have from the study sample themselves, because the third variant does not exist in the external reference panel, so we have to get rid of that. And also the LD is not very accurate if we look at the rare variants, so that's not a good approach. And what we propose to do is that we just multiply this by the random matrix once to get the single variant summary statistics and the stochastic summary statistics U. We get that once, and then we can use any type of variant definition, and then we're basically just extracting the corresponding rows from this U matrix to calculate all the quantities we need. So this is the most flexible way. And you can see that if I'm crazy, I want to look at some distant variant sets, right? I don't want to assume that my variant sets are in close proximity. For example, I want to test this yellow set, but yellow set has 3 regions, right? So you can see that in this way, we will have to, using the traditional approaches, we will have to go back to the individual level data, because otherwise this kind of covariance information was not covered here, right? Because it was not computed or saved. But using our framework, we're basically coloring them with different colors, so we can do this at the meta-analysis stage, so which is very convenient. So here I want to show you the first example is that we try to use the high-chip interaction loops to define the variant sets. So what we're doing here is that from this high-chip data in different tissues, we define the variant sets using these loops. So variants in this left anchor and the right anchor, they were grouped together to do the association test. This is one example from the LDL analysis. It's close to the PCSK9 gene on chromosome number 1. This is a very well-known gene for LDL, but you can see that none of these sliding windows would reach significance. But when you put them together, it's a different story. And this will give you some kind of, it's probably not the best biological example, but at least our tool will enable to do like crazy things like that. This was previously not feasible. It has to be performed using individual level data, even if you have the summary statistics, because these variants may be very far away from each other. But now we can do that. And the study we're using was one of the studies from the Tropp study. So Hispanic community health study. This is a very complex study with 6 Hispanic background groups. So we have a very complex model. We're adjusting for the first ancestry principal components, and we're using 3 random effects to model the relatedness in the study sample. So you can see this was a paper published 8 years ago, looking at the genetic component in the study. So you can see that for most Caribbean, people of the Caribbean ancestry, you can see that they are mostly admixed with the European and the African ancestry, with a very small proportion of American ancestry. Whereas for the continental groups, Mexicans, Central and South Americans, they are mostly admixed with European and American ancestry. So these plots were sorted based on the ascending order of the proportion of global European ancestry, but you can see that there are individuals with almost the same ancestry component on the global level, but if you look at the specific regions, they may still be very different. And this is the famous triangle that you can see that when you have 3 ancestry populations, this is what it looks like. And this plot shows that only the first 5 ancestry PCs were informative, and starting from PC6, they were mostly noise. So that's why we're adjusting for the top 5 PCs. So here's some computational performance. This is a plot showing the p-value from using stochastic summary statistics compared to rerunning the analysis using various changing variant set definitions starting from individual level data analysis. Again, you can see these p-values on the -log10 scale. They were highly concordant. This is a Spearman correlation of those p-values, which is free. It doesn't depend on the monotone function, so it's the same on the raw p-value and the -log10 p-value scale. And on the other hand, you can see that here's a comparison of the CPU time. We can do the analysis, and here's a memory footprint on a laptop. Literally, you can do this on a laptop using stochastic summary statistics. You can decide how you want to play with the data or the summary statistics. You don't have to go back to the individual level data. So this is not feasible, on a laptop. You have to use some computing clusters or cloud. Okay, so now I want to switch gear to talk about the second topic, where the application where we want to compute the LD scores using stochastic summary statistics. So this is a classical way, is to compute the LD scores from the the study samples. So typically we would actually compute the LD first, and then the LD score was defined as the sum of square of all the adjacent genetic SNPs compared to the tag SNP or the index SNP usually within the 1 million base pair window, right? So you have these windows, and then you add them together, you get the LD score. Using the external reference panels, I hope you would agree that if you miss some variants, sometimes this is not because you're using an ancestry unmatched reference panel, even if you have the same ancestry, because usually we're using common variants with minor frequency greater than 1%. If you happen to have a SNP with minor frequency 0.99%, then that's going to be filtered out, right? So you can see that you're losing the LD score for this variant, and this variant, it's inaccurate because you're losing these two, right? For stochastic summary statistics, you only need to compute it once, and then later it's just a matter of fact which rows you want to combine together, right? So you can decide how you want to add them together. So this is very straightforward. So we were comparing them, the LD scores from the study samples. We use two different studies, compare them to the, those computed from external reference panels. We have 3 1000G superpopulations, and you can see that this is a projection matrix for the stochastic summary statistics. This is nothing more than simply taking the genotypes and then subtract by the column mean, right? So you can see that this is an idempotent matrix. Okay, so these are our results. So you can see that on the top left, we're comparing the StocSum computed LD scores with the LDSC LD scores computed from the study sample. So this x-axis was treated as a gold standard because those were the ones that we can compute using individual level data from the study sample. So you can see that we have highly concordant LD scores compared to all the other 7 external reference populations. And surprisingly, you can see that even if you put all of these ancestry populations together, it still didn't work well, right? So the Spearman correlation of the LD scores was less than 0.9. So you can see it doesn't work very well, and it has some consequences. If you try to do LD score regression to estimate heritability, you can see that using StocSum based LD score, you can always get very close results compared to using the LDSC, LD scores. Whereas when you use all of these external reference panels from 1000 genomes, you get quite different heritability estimates. I have to make a disclaimer that because our study sample was quite small, we only have about 7,000 or 8,000 individuals. These heritability estimates for phenotypes are not accurate. But the point is that even though these estimates are not accurate, so our StocSum based estimates are very close to those that were directly computed using the study sample individual level data. So you can see that they are very close. And this shows the computational cost. One major disadvantage of calculating LD scores from the individual level data was that it takes a long time. For longer chromosomes, it can take over 60 CPU days. Even though with prioritization, you can probably finish this within 1 or 2 days for these longer chromosomes, but it's computationally very expensive using the stochastic summary statistics because we just need a matrix multiplication. This is 2 orders of magnitude faster than directly using individual level data. This is only for 7,000 individuals. If you want to scale this up to hundreds of thousands, for example, UK Biobank, then you're going to see much more dramatic difference in terms of the computing time. Okay, so the take-home message here is, number 1, that StocSum-based estimates are more accurate than those using external reference panels. And number 2 is that StocSum-based LD score estimation is the most computationally efficient. Okay, so another question that naturally raised is that, okay, so here you're using the Hispanic Americans, right? So what about European Americans and African Americans? So we look at another study, which is the atherosclerosis risk in community study, the ARIC study. They have European Americans and African Americans. We repeated the same analysis. You can see that the StocSum LD scores were very accurate compared to LDSC on the study samples. But in addition to that, you can see this one also performs pretty well, right? So this is using the European reference panels from 1000G. You can get a high correlation, 0.98. So this works actually pretty well, which is not surprising, right? So you can compare the heritability estimates using the European reference panel. So it's almost similar to what we get. So the take-home message here is that if you use European populations as external reference panel, it performs reasonably well for European Americans. This explains why many external reference panel-based summary statistics methods have been restricted to studies with European ancestry participants, because when you apply them to underrepresented populations, the performance was not good. Okay, so this is my last part. We tried the same analysis using African Americans. You can see that these correlations were not good, because African Americans, these were two-way admixed with European and African ancestry. So their African ancestry, something very interesting here is that they were not fully represented using the African populations from 1,000 genome. Okay, so that's the same conclusion. None of the external reference panels or their combinations performed well for African Americans, which are admixed. And using external reference panels is generally not a good idea for admixed population. I hope you would agree, even if the reference panel cover all the ancestry populations, because when you look at the LD structure, that's really just a local ancestry, and the local ancestry can be very different. Even two individuals have the same global ancestry components. Okay, so we also tried this to look at the genetic correlation, but in the interest of time, I'm going to skip that. Okay, so here's the summary. We have developed a computationally efficient framework for using stochastic summary statistics to represent the cross-variant correlation, or LD structure, from whole genome sequencing studies. And the computational complexity, because this is just a matrix multiplication, they always scales linearly with both the sample size and the number of variants, regardless of the complex statistical model that you're looking at. And so we can allow flexible variant set definition for many under-investigated crazy scientific hypotheses. Now we can test that. And we can also allow flexible meta-analysis to facilitate international collaborations using whole genome sequencing resources. So, you have UK Biobank and all vast data, right? They are hosted on different cloud computing platforms. You cannot combine those data together, but now you can easily combine their stochastic summary statistics and then do something together. And there's no need for external reference panels, and that's extremely important for underrepresented and mixed and isolated populations. And we're hoping that our framework can make genetic research, especially summary statistics-based genetic research, equally beneficial to all human beings. Okay, and finally, I would like to thank, this is the hard work by Dr. Nana Wang, and here are all my collaborators on this project, and I would like to thank Eric and the SOL study participants, Tropp, and support from NIH. Thank you.